Hey everyone, ever wonder how a single cell becomes an entire multicellular organism? Or how some animals can regrow lost body parts? It's all thanks to mitosis, the eukaryotic cell division that creates two identical daughter cells. This happens during the cell cycle, the process of cell growth and division. The G0 phase is when cells perform their main functions. Eye cells see, skin cells protect, muscle cells contract, each doing its job in the body. With the right stimuli, a cell can enter the cell cycle and begin dividing. The first stage is called G1 phase, G for growth or gap in English. During G1, the cell doubles everything except DNA, proteins, organelles, all it needs to divide between daughter cells. During S phase, for synthesis, what's synthesized? DNA. All chromosomes are duplicated. This phase is crucial. It's dedicated to DNA duplication as errors can be fatal for daughter cells. Then comes G2 phase, where we check if everything's okay before cell division begins. During M phase, the actual division of the mother cell into two daughter cells occurs. To recap, G1 phase is for growth, duplicating proteins and organelles. S phase is solely for DNA duplication. Lastly, G2 phase, the important cell cycle checkpoint. And finally, we have the M phase, or mitosis. All phases except M phase are collectively called interphase. Mitosis starts with prophase, where the nucleolus dissolves and chromosomes condense. It's much easier to separate condensed chromosomes than those spread out during interphase. Another crucial step is the breakdown of the nuclear membrane. Clearly, to separate chromosomes, this envelope must be dismantled to move them apart. At this stage, a chromosome is split into two sister chromatids. Each chromatid is a copy, but together they still form one chromosome. Another prophase event is cell polarization due to centrioles. Each centriole moves to a cell pole and generates aster fibers. During metaphase, centrioles create spindle fibers that align chromosomes in the cell's middle, forming the equatorial plate. Think, with two poles, what line passes through the cell's center? The equator line. That's the equatorial plate. Anaphase is the actual separation phase. Mitotic spindles shorten, pulling apart chromosomes and separating chromatids at the centromere. Each chromatid moves to one side, becoming a chromosome in the daughter cell. Finally, telophase, the separation phase where the cell elongates, narrows in the middle, and splits into two daughter cells, each with a centriole and a chromatid from each original chromosome. In telophase, the nuclear membrane reforms, nucleoli reappear, and chromosomes decondense to their original state. Each daughter cell is then identical to the mother cell that started the process.